Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam
Eh,
warahmatullahi wabarakatuh.
Selamat pagi semuanya. Good morning everyone, especially Dr. Warrick and Mr. Jeff Chen. Nice to meet you. Terima kasih banyak kepada moderator eh, dan juga PT Sciencewerke atas kesempatan yang diberikan kepada saya untuk bisa berbagi dalam webinar ini. Perkenalkan, saya Yeni Wahyuni Hartati dari Departemen Kimia dan Pusat Studi Sensor dan Kimia Hijau, seperti tadi telah disampaikan oleh moderator Fakultas MIPA UNPAD. Hari ini saya akan membahas topik yang sangat relevan dengan perkembangan teknologi kesehatan saat ini, yaitu Introduction to Biosensors and their Applications in Point of Care Testing atau sering disingkat POCT. Mungkin tanpa kita sadari, beberapa dari kita sudah pernah menggunakan teknologi ini, misalnya alat cek gula darah atau rapid test ya. Dan di balik alat-alat sederhana tersebut sebenarnya ada konsep ilmiah yang sangat menarik dan terus berkembang. Jadi hal yang melatar belakangi mengapa teknologi ini menjadi sangat penting, yaitu jika kita melihat metode diagnostik konvensional yang selama ini digunakan, misalnya teknik PCR atau ELISA ya. Metode tersebut memang memiliki akurasi yang tinggi, namun ada beberapa keterbatasan utama ya. Pertama, waktu analisis yang relatif lama, mulai dari pengambilan sampel, preparasi hingga hasil keluar itu bisa memakan waktu berjam-jam bahkan berhari-hari. Kedua, tentu saja biaya yang tidak murah, baik dari sisi alat maupun operasional ya. Dan ketiga, metode ini umumnya memerlukan fasilitas laboratorium dengan peralatan yang kompleks ya, dan tenaga ahli yang terlatih. Nah, di sisi lain, kebutuhan di lapangan itu justru mengarah ke hal yang sebaliknya. Kita membutuhkan sistem diagnostik yang cepat, bisa dilakukan langsung di lokasi, portable, dan dengan biaya yang terjangkau. Terutama dalam kondisi seperti daerah terpencil, situasi darurat, atau bahkan untuk monitoring kesehatan secara mandiri. Nah, disinilah point of care atau POCT berbasis biosensor hadir sebagai solusi ya. Jadi teknologi ini memungkinkan deteksi dilakukan secara langsung di dekat pasien dengan waktu yang jauh lebih singkat, alat yang lebih sederhana, dan tetap mempertahankan sensitivitas yang baik. Sehingga keputusan klinis dapat diambil lebih cepat dan tepat ya. Ini menjadi salah satu alasan mengapa riset di bidang biosensor itu berkembang sangat pesat saat ini. Nah, secara sederhana, biosensor ini dapat didefinisikan sebagai suatu perangkat analitik yang mampu mengubah respon biologi atau respon biologis menjadi sinyal yang dapat diukur. Artinya, biosensor bekerja dengan menerjemahkan interaksi biologis, misalnya antara antigen dengan antibodi atau DNA dengan pasangan komplementernya, menjadi sinyal yang bisa kita baca seperti sinyal listrik atau perubahan warna atau pembacaan lainnya ya, bisa spektrum, kemudian bisa juga angka. Kalau kita lihat lebih dalam, biosensor itu terdiri dari tiga komponen utama. Yang pertama adalah bioreseptor di sini, yaitu bagian yang bertanggung jawab untuk mengenali target secara spesifik. Yang kedua adalah transducer, yang berfungsi mengubah interaksi biologis antara target dan bioreseptor menjadi sinyal yang terukur. Dan yang ketiga adalah sistem pemrosesan sinyal yang akan menampilkan hasil dalam bentuk yang mudah dipahami oleh user ya atau pengguna. Yang menarik, kombinasi dari ketiga komponen ini memungkinkan kita untuk melakukan deteksi tadi yang cepat, spesifik, bahkan bisa dilakukan secara real time. Inilah yang membuat biosensor menjadi teknologi kunci dalam pengembangan biosity ya. Secara umum, prinsip kerja biosensor dapat dibagi menjadi tiga tahapan. Pertama, pengenalan atau recognition. Di sini bioreseptor akan berinteraksi secara spesifik dengan target analit. Misalnya antibodi akan mengenali antigen, seperti tadi ya DNA akan mengenali pasangannya atau molekul tertentu. Interaksi ini sangat spesifik sehingga menjadi dasar utama dari akurasi biosensor. Tahap kedua adalah transduksi sinyal. Interaksi biologis tadi tidak akan berlangsung, tidak bisa kita lihat atau kita ukur, sehingga perlu dikonversi menjadi sinyal fisik atau kimia yang terukur yang bisa dibaca. Inilah peran transducer. Dan tahap ketiga adalah pemrosesan atau pembacaan sinyal. Sinyal yang dihasilkan kemudian diproses dan ditampilkan dalam bentuk yang mudah dipahami, misalnya tadi angka atau spektrum atau gerakan jarum gitu ya. Jadi kalau dilihat dari sisi yang lebih mendalam, terutama untuk biosensor elektrokimia, proses ini lebih melibatkan ke fenomena di permukaan elektrode ya nanti. Nanti mungkin akan dibahas. Dan biosensor ini diklasifikasikan menjadi beberapa di antaranya berdasarkan bioreseptor ya. Bioreseptor itu merupakan otaknya dari biosensor karena bagian inilah yang menentukan selektivitas. Apakah sensor mampu mengenali target secara spesifik atau tidak gitu. Ada beberapa bioreseptor yang penting, di antaranya enzim. Enzim ini banyak digunakan karena memiliki aktivitas katalitik yang tinggi. Contohnya dalam eh, glukometer, dalam sensor glukosa ya. Ya, enzim glukooksidase pada biosensor glukosa itu mengkatalisis reaksi yang mengkatalisis reaksi glukosa ya oksidasi glukosa menghasilkan sinyal yang dapat diukur. Kemudian kedua antibodi yang digunakan dalam imunosensor karena dikenal antibodi itu sebagai senyawa imun. Interaksi antigen dan antibodi sangat spesifik sehingga cocok untuk deteksi biomarker penyakit ya. Dan ketiga asam nukleat seperti DNA atau RNA. Jenis ini banyak digunakan untuk deteksi genetik, misalnya pada patogen atau DNA lainnya gitu ya selain patogen untuk identifikasi melalui mekanisme hibridisasi. Selanjutnya ada aptamer. Aptamer itu oligonukleotida sintetis yang dapat dirancang untuk mengikat target tertentu. Keunggulan aptamer adalah lebih stabil dan dapat diregenerasi dibanding antibodi. Jadi prinsipnya interaksinya sama afinitas gitu ya. Tetapi kalau aptamer itu oligonukleotida, kalau antibodi itu tadi apa proteinnya. Dan metode terkini ini adalah CRISPR base ya atau CRISPR yang menawarkan kemampuan pengenalan target dengan spesifisitas yang sangat tinggi, bahkan dengan mekanisme amplifikasi sinyal secara intrinsik. Namun selain uh, klasifikasi atau pengelompokan berdasarkan bioreseptor ini, biosensor itu diklasifikasikan juga berdasarkan transducernya ya. Klasifikasi ini uh, yang umum dan sebetulnya ini banyak sekali dikembangkan untuk POCT itu adalah transducer elektrokimia ya. Ini merupakan jenis yang eh, paling banyak dalam POCT. Sinyal yang dihasilkan bisa berupa arus ya dalam voltammetrik atau amperometric method. Kemudian potensial dalam sistem potensiometri dan impedansi dalam sistem impedometrik. Keunggulannya adalah sensitivitasnya tinggi, sistemnya sederhana dan mudah untuk diminiaturisasi ya. eh, eh, Sistem elektrokimia itu. Yang kedua adalah sistem optik ya. Jenis ini bekerja dengan mendeteksi perubahan sinyal optik seperti absorbansi, fluoresensi, fenomena surface plasmon resonance ya, atau SPR. Contohnya dalam POCT yang paling umum adalah jenis lateral flow assay yang sering digunakan pada rapid test. Jadi yang dilihat warnanya ya atau kolorimetri. Kemudian yang ketiga adalah biosensor piezoelektrik. Sensor ini mendeteksi perubahan massa pada permukaan yang menyebabkan perubahan frekuensi resonansi seperti QCM atau quartz crystal microbalance ya. Dan ada juga biosensor thermal ya, yang mengukur perubahan panas menjadi panas ya akibat reaksi biokimia dari bioreseptor dengan target. Meskipun penggunaan ini lebih terbatas ya yang biosensor thermal ini. Jadi pemilihan jenis transducer itu tidak hanya bergantung pada target analit, tapi juga pada kebutuhan sensitivitas dan kompleksitas sistem dan aplikasi pada akhirnya ya. Dan klasifikasi yang ketiga adalah berdasarkan target analitnya ya. Klasifikasi ini sangat penting karena setiap jenis analit memiliki karakteristik yang berbeda, sehingga memerlukan strategi dalam pengembangannya dan sangat spesifik ya. Jadi ada target analit yang berbasis metabolit ya, seperti glukosa, kemudian laktat, kolesterol, asam urat. Biosensor biosensor metabolit ini biasanya gitu banyaknya ya yang sudah dikomersialisasi itu berbasis enzim karena melibatkan reaksi katalitik yang menghasilkan sinyal yang terukur. Kemudian ada juga biosensor patogen ya. Ini patogen ini seperti bakteri dan virus. Pendekatannya bisa berbasis antigen atau DNA tadi ya, genetik atau bahkan whole cell detection. Target biomolekul seperti protein ya, enzim atau biomarker penyakit. Ada banyak biomarker penyakit yang merupakan senyawa protein. Seperti tadi untuk mendeteksi biomarker-biomarker kanker yang tadi disampaikan sama moderator ya, itu beberapa atau banyak dari jenis protein ya. Kemudian biomolekul ini selain protein juga ada yang berbasis DNA tadi ya, karena untuk mendeteksi biomarker penyakit ada juga yang berbasis DNA, kemudian ada juga yang karbohidrat misalnya ya, atau senyawa lipid. Kemudian juga ada biosensor logam berat ya, heavy metal. Jadi untuk aplikasi lingkungan dan termasuk toksin ya, ini senyawa kontaminan, zat kimia dan kontaminan lingkungan. Ini seperti pestisida juga sering digunakan aptamer ya umumnya sebagai bioreseptornya. Atau material fungsional lainnya seperti yang banyak dikembangkan dan kami kembangkan juga eh, bioreseptor sintetis ya, seperti molecularly imprinted polimer ya, polimer yang dicetak sesuai molekul apa yang mau kita deteksi. Jadi jika kita lihat lebih dalam, tantangan utama dalam klasifikasi berbasis target ini adalah bagaimana mencapai selektivitas dalam matriks sampel yang kompleks gitu ya, seperti sampel darah, air limbah misalnya, atau bahkan sampel makanan. Oleh karena itu, desain biosensor harus mempertimbangkan tidak hanya target gitu, tapi juga kondisi nyata dari sampel yang kita analisis. Nah, dengan berbagai jenis target yang tadi bisa dideteksi, pertanyaannya adalah bagaimana kita membawa teknologi ini keluar dari laboratorium gitu, dan menggunakannya langsung di lapangan. Disinilah konsep POCT tadi ya, point of care testing, menjadi sangat krusial atau penting. Konsep POCT ini berkembang dengan mentransformasikan biosensor menjadi perangkat yang portable, cepat, dan mudah digunakan bahkan oleh non-ahli gitu ya, oleh umum orang. Kunci utama dari transformasi ini adalah miniaturisasi. Melalui miniaturisasi ini, sistem biosensor dikonversi menjadi perangkat yang handheld, bisa dipegang gitu ya, seperti glukometer, pH meter, dan perangkat portable lainnya. Ini memungkinkan analisis dilakukan secara langsung di lapangan dengan alat yang sederhana, portable, mudah dibawa-bawa gitu, dan hasilnya cepat. Selain itu, kita juga melihat integrasi dengan platform mikrofluidik, baik berbasis kertas, gelas, atau kaca maupun polimer ya. Mikrofluidik ini memungkinkan pengelolaan sampel dalam volume kecil secara otomatis melalui gaya kapiler tanpa memerlukan pompa eksternal. Hal ini memberikan beberapa keuntungan utama, di antaranya mengurangi kebutuhan volume sampel, mempercepat waktu analisis, dan meningkatkan efisiensi dan portabilitas dari sistem. Jadi miniaturisasi ini merupakan apa ya, untuk menyampaikan dari lab ke lapangan tadi ya. Nah, bagaimana biosensor berkembang menjadi POCT ini? Pertanyaan berikutnya adalah seperti apa perangkat POCT yang ideal? Nah, untuk menjawab ini, WHO ini memperkenalkan konsep yang sangat terkenal yaitu kriteria assured ya. Jadi E-nya affordable, artinya perangkat harus terjangkau sehingga dapat digunakan secara luas, termasuk di negara berkembang. Kemudian S-nya sensitif, harus mampu mendeteksi target dalam konsentrasi yang rendah, gitu. Kemudian S yang kedua spesifik, harus selektif, spesifik, hanya merespon target yang diinginkan. Ya namanya sensor ya harus menuju target itu gitu ya. Dalam matriks yang kompleks. U-nya user friendly, mudah digunakan bahkan tadi oleh non ahli atau non tenaga medis. Kemudian R-nya rapid and robust. Ini hasilnya harus cepat dan sistem harus stabil dalam berbagai kondisi lingkungan. Kemudian equipment free sebisa mungkin tidak bergantung pada alat tambahan yang kompleks. Jadi tadi dibuat mini dan portable ya. Kemudian D-nya deliverable to end users. Artinya mudah didistribusikan dan benar-benar sampai ke pengguna yang membutuhkan. Jadi tadi di daerah terpencil, di puskesmas, di apa bisa bisa terjangkau, bisa digunakan, tidak harus ke kota misalnya untuk mengecek suatu atau dalam hal ini misalnya untuk diagnosis awal gitu ya, diagnosis dini. Jadi diagnosis yang diharapkan itu tidak hanya akurat tetapi juga accessible gitu. Dan menariknya biosensor itu terutama yang tadi terintegrasi VOCT ini dirancang untuk memenuhi hampir semua kriteria assured ini. Nah, platform biosensor yang banyak dikembangkan untuk VOCT ini diantaranya atau yang paling banyak seperti tadi saya sampaikan paling dominan adalah eh, platform elektrokimia ya. Mengapa elektrokimia? Mengapa elektrokimia? Karena teknologi ini menawarkan kombinasi yang sangat ideal ya. Sensitivitasnya tinggi, biayanya rendah, dan kemudahan diminiaturisasi. Jadi secara prinsip, biosensor elektrokimia bekerja dengan mengubah interaksi biokimia menjadi sinyal listrik. Sinyal ini bisa berupa arus, potensial, atau perubahan impedans yang kemudian dikorelasikan dengan konsentrasi analit. Jadi tiga mode utama yang sering digunakan tuh voltametrik atau amperometrik, di mana kita mengukur arus yang dihasilkan dari reaksi redoks. Contohnya adalah biosensor glukosa ya, di mana enzim glukooksidasi itu akan menghasilkan hidrogen peroksida yang kemudian dioksidasi di permukaan elektroda. Yang kedua, metodenya tuh potensiometrik, pengukurannya ya mengukur perubahan potensial akibat interaksi ionik. Dan uh, yang ketiga itu impedimetrik, jadi atau ACE yang sangat sensitif terhadap perubahan resistensi di permukaan elektroda. Misalnya ketika target berikatan akan terjadi peningkatan resistensi transfer muatan, bisa kita lihat pada plot uh, naik Q-nya. Nah, di sisi materialnya untuk pengembangan elektrokimia biosensor ini sangat mempengaruhi bagaimana kinerja sensor itu ya atau biosensor itu. Jadi permukaan elektrode biasanya dimodifikasi dengan nanomaterial yang umum misalnya emas nanopartikel ya, grafen atau turunan karbon atau MOPS atau tadi MIPS ya. Permukaannya menjadi lebih aktif dan mempercepat transfer elektron. Metode modifikasinya bermacam-macam ya, ada teknik elektrodeposisi, ada teknik broadcast, ada teknik spinning dan lain-lain. Kemudian platform ini sangat kompatibel dengan teknologi SPE ya, screen print electrode yang memungkinkan produksi massal dengan biaya yang rendah. Jadi inilah yang membuat biosensor elektrokimia uh, sangat cocok untuk POCT karena dibuat kecil, murah, tetapi tetap sensitif gitu ya. Uh, jadi banyak contoh tadi yang sudah disebutkan juga seperti tadi glukosensor ya, kemudian sensor berbasis CRISPR juga sudah banyak dikembangkan. Nah, sensor elektrokimia ini pada perangkat POCT bisa digunakan untuk menganalisis berbagai biofluid, sampel biologis ya, yang mencerminkan kondisi fisiologis tubuh. Jadi misalnya dari sampel darah yang merupakan biofluida utama karena kaya informasi. Namun pengambilannya bersifat invasif, jadi harus diambil darah ya. Sehingga mendorong penggunaan alternatif non-invasif ini seperti cairan interstisial, keringat, saliva, dan air mata juga menjadi salah satu alternatif ya. Biofluida non-invasif ini memungkinkan pemantauan kesehatan secara lebih nyaman ya dan real time. Meskipun beberapa seperti air mata masih menghadapi tantangan teknologi agak susah mengambil sampelnya. Jadi secara umum teknologi ini fleksibel dan dapat disesuaikan untuk berbagai aplikasi diagnostik dengan memanfaatkan beragam jenis biosensor seperti bioreseptor ya seperti enzim, antibodi dan aptamer tadi. Jadi contoh yang paling sukses yang tadi banyak disebutkan dari POCT ini adalah eh, ada sensor glukosa ya atau glukosa biosensor. Ini telah merevolusi manajemen diabetes di seluruh dunia ya. Biosensor ini umumnya berbasis enzim yaitu glukooksidase yang memiliki selektivitas tinggi terhadap glukosa. Jadi secara mekanisme prosesnya dimulai ketika glukosa dari sampel berinteraksi dengan enzim glukooksidase. Kemudian reaksi yang terjadi ada oksidasi glukosa menjadi asam glukonat dengan oksigen sebagai aseptor elektron menghasilkan hidrogen peroksida ya. Selanjutnya hidrogen peroksida ini akan mengalami oksidasi di permukaan elektroda menghasilkan elektron yang kemudian diukur sebagai arus yang berbanding lurus dengan konsentrasi glukosa. Jadi ini tahapan reaksinya ya. Dan arus yang dihasilkan dari proses oksidasi tadi akan berbanding lurus tadi dengan konsentrasi ya. Sehingga kalau dalam sistem yang lebih modern sering digunakan redox mediator supaya tidak bergantung dari oksigen tadi ya. Hal ini membantu menurunkan overpotensial dan meningkatkan sensitivitas. Dari sisi elektrokimia, mekanisme ini termasuk dalam jenis voltametri atau amperometri ya, potensialnya dibuat tetap. Di mana kita mengukur arus sebagai fungsi dari konsentrasi analit. Jadi tadi berbanding lurus ya, harus berbanding lurus dengan konsentrasi. Nah, performa sensor ini dipengaruhi oleh beberapa faktor, kinetika enzim, kemudian difusi substrat ke permukaan elektrode, dan efisiensi transfer elektron di permukaan elektrode. Jadi penggunaan nanopartikel banyak dikembangkan ya untuk apa meningkatkan terus gitu improvement di sensor ini juga material-material lainnya ya. Ini yang membuat biosensor glukosa ini memiliki respon yang cepat, sensitivitasnya tinggi dan stabilitas yang cukup baik untuk penggunaan sehari-hari. Jadi karena keberhasilannya dari biosensor glukosa ini sering dianggap sebagai benchmark ya dalam pengembangan biosensor lainnya. Dan prinsip yang sama itu kini banyak diadaptasi untuk berbagai aplikasi lain termasuk deteksi biomarker atau tadi penyakit infeksi ya. Jadi itu contoh yang paling banyak dari electrochemical biosensor untuk POCT ya. Nah, selain berbasis elektrokimia, tadi ada biosensor optis ya, atau optical biosensor. Ini berbeda dengan elektrokimia yang menghasilkan sinyal listrik. Biosensor optik ini bekerja dengan mendeteksi perubahan sifat optik seperti warna, intensitas cahaya, dan atau panjang gelombang ya. Atau tadi surface Fresnel resonance ya. Pendekatan yang paling sederhana dan umum ini digunakan adalah deteksi kolorimetri ya seperti dalam lateral flow, dalam lateral flow assay yang atau rapid test. Ketika target muncul, akan muncul garis berwarna yang menunjukkan hasil yang positif. Selain itu juga banyak dikembangkan untuk POCT yang berbasis optik ini adalah yang biosensor fluoresensi ya, di mana sinyal dihasilkan dari emisi cahaya setelah eksitasi. Metode ini umumnya memiliki sensitivitas yang lebih tinggi dibandingkan kolorimetrik, namun membutuhkan alat pembaca gitu ya. Eh, tetapi karena tadi untuk Curiosity, alat pembacanya juga dibuat sederhana. Jadi dari sisi mekanismenya, eh, pengikatan target oleh bioreseptor, kemudian aktivasi label optik, misalnya nanopartikel emas ya atau fluorofor, dan konversi menjadi sinyal visual atau optis. Keunggulannya sederhana, cepat, seperti halnya keunggulan velocity pada umumnya. Dapat memberikan hasil secara langsung ya, visual readout. Sangat cocok untuk screening di lapangan. Namun ada juga keterbatasannya yaitu sensitivitasnya bisa lebih rendah dibanding metode elektrokimia atau molekuler lainnya ya. Dan dalam beberapa kasus, interpretasi visual bisa bersifat subjektif ya. Nah, untuk mengatasi hal ini banyak dikembangkan integrasi dengan smartphone ya. Eh, jadi smartphone based detection ya, di mana kamera digunakan untuk meningkatkan akurasi pembacaan sinyal. Jadi bisa dikatakan bahwa biosensor optis ini sangat ideal untuk aplikasi POCT ya, eh, yang membutuhkan kesederhanaan, kecepatan, terutama ketika sumber dayanya terbatas. Kemudian contoh aplikasi yang lebih kompleks seperti deteksi tadi, deteksi penyakit ya, dengan dua pendekatan utama yang umum digunakan dalam POCT itu yang antigen based detection dan nucleic acid based detection. Keduanya memiliki prinsip keunggulan dan keterbatasan yang berbeda. Jadi kalau berbasis antigen, pendekatan pertama adalah eh, ini tadi yang banyak digunakan di rapid test ya, mekanismenya sederhana. Terbentuk kompleks antigen antibody, kompleks ini menghasilkan warna. Kemudian yang pendekatan berbasis asam nukleat atau DNA ini mekanismenya melibatkan proses hibridisasi ya, hibridisasi antara probe dengan target. Atau menggunakan sistem CRISPR, jadi tanpa amplifikasi di dalam sampel, udah langsung menuju ke situs atau bagian yang dikenali oleh uh, Cas sistem ya, CRISPR-Cas atau kompleks antara enzim Cas dengan guide guide RNA-nya atau sistem CRISPR-nya. Tetapi dalam banyak kasus banyak digunakan juga teknik amplifikasi PCR ya atau isothermal amplification atau LAMP. Hasil deteksi biasanya berupa sinyal fluoresensi atau sinyal elektrokimia gitu. Jadi untuk eh, deteksi penyakit ini banyak dikembangkan baik secara elektrokimia maupun secara optis ya. Jadi mekanismenya itu eh, antara antigen antibody, kalau dalam sistem antigen antibody itu bisa sinyal itu bisa berasal dari binding event langsung, sedangkan pada asam nukleat melibatkan sinyal amplifikasi baik melalui enzim atau sistem CRISPR tadi ya. Kemudian ada lagi POCT yang dikembangkan berupa wearable dan smartphone based biosensor ya. Jadi yang berkembang tidak hanya menjadi perangkat POCT tapi juga terintegrasi di kehidupan sehari-hari ya untuk wearable ini. Jadi ini wearable sensor ini mampu menganalisis berbagai jenis cairan biologis tadi seperti keringat, saliva, cairan interstisial. Melalui pendekatan ini kita melakukan monitoring biokimia secara non-invasif ya, dan continue, bisa real time juga. Jadi sebagai contoh, sensor keringat dapat digunakan untuk memantau elektrolit misalnya atau metabolit, sementara sensor cairan interstisial banyak dikembangkan untuk monitoring glukosa secara real time ya, bisa juga. Kemudian sistem integrasi dengan smartphone tadi tidak hanya berfungsi sebagai alat pembaca, tapi juga sebagai pusat pemrosesan data dan konektivitas ya. Dengan bantuan kameranya, kita bisa melakukan deteksi berbasis warna atau intensitas sinyal, sementara aplikasi mobilenya itu memungkinkan analisis data secara langsung ya, tadi secara real time dan bisa dihubungkan ke sistem cloud ya. Ini menjadi menjadi lebih luas ya aplikasinya. Kemudian ini monitoring kesehatan yang berkelanjutan ya, ini jadi teknologi ini menjadi penting ya dalam digital health. Kemudian manajemen penyakit kronis hingga pengembangan personalized medicine ini untuk insight kesehatan tadi yang real time dan accessible tadi ya, actionable juga. Nah, ini beberapa contoh aplikasi dari wearable biosensor ya. Yang ini tuh perangkat pemantauan glukosa berbasis keringat yang bersifat wearable dan disposable, dilengkapi dengan modul penghantaran obat transdermalnya ya, yang secara bertahap. Jadi ini ditempel di tangan misalnya. Kemudian ini representasi skematik lensa kontak pintar ya, yang dapat dikendalikan dari jarak jauh untuk pemantauan glukosa secara non-invasif. Jadi pemantauan glukosa yang diaplikasikan atau ditempelkan diterapkan di lensa kontak. Kemudian ada lagi biosensor tato ya, untuk pemantauan elektrokimia laktat pada permukaan kulit. Diaplikasikan pada otot. Biasanya ini percobaannya pada seorang relawan pria gitu ya. Kemudian ada lagi sistem wearable itu berbasis osmotik terintegrasi untuk deteksi laktat dalam keringat ya. Terdiri dari hidrogel osmotik, mikrofluida basis kertas, sensor laktat terfungsionalisasi serta potentiostat yang fleksibel dan nirkabel ya. Kemudian ada juga ini biosensor elektrokimia berbasis elektroda cetak atau screen elektrod tadi yang dimodifikasi dengan karbon ya, di percobaannya ini dimodifikasi dengan uh, nanowalks karbon untuk mendeteksi asam urat ya, dimodifikasi pakai urikase, enzim urikase. Kemudian ini ada juga biosensor berbentuk mouth guard ini dilengkapi dengan rangkaian amperometrik yang nirkabel dan elektrode kerja karbon berbasis Prussian blue yang dimodifikasi secara kimia serta dimobilisasi dengan urikase juga ya untuk asam urat. Nah, ini skema bagaimana misalnya suatu screen print electrode dimodifikasi. Jadi ini ada tahapan modifikasi step by step. Contohnya ini untuk mendeteksi secara kimia protein yang kaya dengan histidin dari suatu organisme ya, suatu patogen Plasmodium falciparum ya, dalam sampel darah. Jadi si working electrode ini pertama dimodifikasi uh, dengan nanomaterial misalnya, kemudian dengan antibodi pertama yang mengikat reporter dalam hal ini metilen biru ya. Ini jadi ini karena reaksinya couple, jadi ini tadi bukan nanomaterial ya. Tapi umumnya si elektroda pertama dimodifikasi dengan nanomaterial kalau misalnya untuk meningkatkan kinerja tadi ya. Kalau ini karena sistemnya pakai mediator redox ya, kompleks rutenium dan nanti dia akan mengalami reaksi redox dengan metilen biru. Dan ini setelah modifikasi yang bertahap tadi baru diteteskan sampel, misalnya ini sampel darah ya. Kemudian yang di bawah ini skema perangkat potensiostat portable yang diintegrasikan dalam POCT ya. Perangkat ini mampu melakukan pengukuran biomarker seperti kortisol di permukaan kulit ya dan laktat secara langsung dan cepat. Nah, selain yang wearable tadi, selain elektrokimia, kemudian optical, wearable. Ada juga paper-based biosensor yang terintegrasi untuk mikrofluidik ya. Ini merupakan salah satu platform yang sangat menjanjikan juga untuk POCT. Jadi prinsip utamanya dari teknologi ini sangat sederhana, namun powerful, yaitu memanfaatkan gaya kapiler alami dari kertas untuk mengalirkan sampel tanpa memerlukan pompa eksternal. Jadi struktur kertas yang berpori memungkinkan cairan bergerak secara pasif melalui saluran yang mikro yang biasanya dibuat menggunakan teknik yang sederhana ya, teknik wax printing atau screen printing di permukaan kertas. Ini sering disebut micropad ya, microfluidic paper-based analytical devices. Eh, Sistem ini memiliki keunggulan ya, disposable tadi sekali pakai dan tentu saja faktor lingkungan juga bisa diperhatikan ya. Biayanya sangat rendah dan ini tidak memerlukan sumber daya listrik eksternal, juga mudah diproduksi secara massal. Dan tadi bisa accessible ya, didistribusikan ke daerah yang terpencil. Jadi aplikasi biosensor yang tadi kita bicarakan itu umumnya dalam bidang diagnostik klinik ya, atau clinical diagnostic. Nah, selain itu POCT berbasis biosensor, biosensor yang dikembangkan untuk POCT juga bisa digunakan untuk pemantauan lingkungan, untuk sampel-sampel seperti tadi ya, logam berat, pestisida, toksin dalam air dan tanah secara cepat dan online juga, dengan prinsip seperti tadi bisa kembangkan, sangat bisa gitu. Kemudian analisis keamanan uh, pangan juga ya. Di sektor ini biosensor berperan untuk mendeteksi patogen, residu kimia atau bahan berbahaya atau autentifikasi ya, pemalsuan makanan gitu ya. Kami juga mengembangkan misalnya untuk halal gitu dan lain-lain. Untuk membantu menjamin keamanan konsumsi. Dan juga di bidang veteriner ya, peternakan dan pertanian, misalnya untuk deteksi penyakit pada hewan ternak atau monitoring kualitas tanah dan juga hasil pertanian. Jadi meskipun aplikasinya sangat beragam, prinsip dasarnya sama gitu, yaitu deteksinya tuh inginnya cepat, spesifik, dan dapat dilakukan di lokasi. Hal ini menunjukkan bahwa biosensor VOCT ini bukan hanya teknologi untuk kesehatan manusia, tetapi juga memiliki peran yang luas dalam mendukung kesehatan dan lingkungan. Nah, beberapa tantangan utama dalam pengembangan biosensor ini, khususnya untuk aplikasi POCT ini, pertama, stabilitas bioreseptor ya. Ini masih menjadi isu penting. Komponen biologis seperti enzim, tadi antibodi dapat mengalami degradasi akibat suhu atau penyimpanan gitu ya, pH, sehingga mempengaruhi kinerja biosensor. Kemudian juga yang kedua, tantangannya adalah gangguan matriks sampel. Biofluida seperti darah, salivak, keringat mengandung banyak komponen lain yang dapat mengganggu interaksi antara bioreseptor dengan target ya, sehingga dapat menurunkan akurasi dan selektivitas deteksi. Kemudian juga reproducibility dan scalability ya. Ini menjadi tantangan. Sensor yang bekerja baik di laboratorium belum tentu memberikan hasil yang konsisten saat diproduksi massal atau digunakan di lapangan. Dan yang terakhir ini isu regulasi ya. Seringkali menjadi hambatan dalam komersialisasi. Jadi biosensor harus melalui proses validasi dan persetujuan yang ketat untuk memastikan keamanan dan keandalannya gitu ya, sebelum dapat digunakan secara luas. Jadi meskipun tadi biosensor ini sangat menjanjikan, berbagai tantangan ini perlu diatasi untuk memastikan implementasi yang optimal di dunia nyata. Dan arah perkembangan biosensor yang semakin inovatif itu tadi ya, di pengembangan biosensing berbasis CRISPR ya. Ini memungkinkan deteksi material genetik dengan tingkat spesifisitas dan sensitivitas yang sangat tinggi dan tidak perlu amplifikasi dengan PCR gitu. Bahkan untuk konsentrasi yang sangat rendah juga bisa terdeteksi tadinya tidak perlu amplifikasi. Yang kedua, eh, ini apa kecerdasan buatan ini, ini dulu deh, nanomaterial. Nanomaterial seperti nanopartikel emas, grafen, dan MOPS ini memberikan peningkatan performa sensor melalui luas permukaan yang besar ya dan sifat listrik yang unggul. Kemudian sistem yang multiplex ya. Ini memungkinkan analisis beberapa biomarker sekaligus dalam satu pengujian sehingga lebih efisien lagi gitu ya, lebih efisien dan komprehensif. Juga telah dilaporkan sistem yang rasiometri ya. Jadi kalau misalnya biomarker A naik, biomarker B turun seperti untuk pengujian kolesterol ya LDL dan HDL gitu. Kalau HDL tinggi, LDL rendah, nah itu sekaligus gitu ya. Jadi bisa multiplex. Kemudian tadi kecerdasan buatan ya atau AI ini membantu dalam pengolahan dan interpretasi data secara otomatis ya. Sehingga meningkatkan akurasi dan kecepatan diagnosis. Dan yang terakhir itu teknologi lab on chip. Ini mengintegrasikan seluruh proses analisis dalam satu perangkat miniatur. Sehingga mendukung diagnosis yang cepat dan portable di prinsip POCT ya. Dan prospeknya ini ke depannya biosensor ini akan memainkan peran penting dalam transformasi sistem kesehatan dengan tadi memperhatikan tantangan-tantangan tadi yang terus berkembang untuk diatasi ya. Salah satunya adalah menuju personalized medicine ini di mana diagnosis dan terapi dapat disesuaikan dengan kondisi spesifik setiap individu ya. Kemudian biosensor tadi yang memungkinkan untuk real-time health monitoring, sehingga kondisi pasien dapat dipantau secara kontinu tanpa harus selalu ke fasilitas kesehatan. Hal ini mendukung perkembangan sistem kesehatan yang desentralisasi, ya terdesentralisasi, di mana diagnosis ini dapat dilakukan di rumah atau di komunitas, tidak hanya di laboratorium atau di rumah sakit. Dan lebih lanjut, integrasi dengan IoT dan platform digital memungkinkan data kesehatan itu dikirim dengan cepat, dianalisis dengan cepat, dan disimpan secara real time yang membuka peluang besar dalam telemedicine dan digital health. Jadi kesimpulannya, biosensor memiliki peran yang sangat penting ya dalam pengembangan point of care testing atau POCT. Teknologi ini menawarkan keunggulan utama berupa diagnosis tadi yang cepat, sensitif, dan portable yang tadi sering disebutkan. Sehingga sangat relevan untuk kebutuhan kesehatan modern. Dengan berbagai inovasi yang terus berkembang, biosensor memiliki potensi besar untuk merevolusi sistem layanan kesehatan di masa depan menuju diagnosis yang lebih cerdas, terintegrasi, dan berbasis kebutuhan tiap individu. Demikian presentasinya. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh. Ya.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih banyak Prof Yeni atas pemaparan materinya yang sangat detail dari mulai basic sampai dengan eh, aplikasi, kemudian juga mengenai apa saja halang rintangan yang mungkin dihadapi para peneliti ataupun pengembang biosensor. Dan ini sangat memberikan inspirasi kepada semua peserta webinar ya. Mungkin karena Prof Yeni juga ini adalah kepala dari Pusat Studi Sensor dan juga Kimia Hijau. Mungkin jika Bapak Ibu ingin berdiskusi lebih detail mengenai ini mungkin bisa menghubungi Prof Yeni ya, Prof.
Ya, iya. Saya
Ya,
stop share ya.
ya, baik, Prof.
Terima kasih.
Ya, tadi kita sudah mendengarkan mengenai bagaimana basic teori dari biosensor itu seperti apa, kemudian bagaimana biosensor dibuat di laboratorium, kemudian juga aplikasinya yang ternyata biosensor itu tidak hanya bisa digunakan untuk pendeteksian penyakit gitu, tetapi juga bisa digunakan untuk lebih banyak lagi aplikasi termasuk monitoring lingkungan, kemudian tadi apa namanya bisa digunakan juga di pertanian, kemudian ada di peternakan dan lain-lain seperti itu. Lalu kemudian selanjutnya adalah bagaimana ketika semua itu sudah dilakukan di laboratorium, lalu kita akan memproduksinya dalam skala yang besar, seperti itu. Nah, eh, ini akan disampaikan oleh pembicara selanjutnya, yaitu Dr. Warrick.
Good morning, everyone. I would like to share the slide first.
Yeah, I will introduce you first, Dr.
Okay,
Warwick.
okay, okay,
Yeah,
okay.
yeah.
Sorry.
So, yeah, Dr. Warwick Su is a chairman of the PICS Corporation and executive director of M.Si Biocorp in Taiwan. And Dr. Warwick Su is a scientist and also technology executive with like 5 years more international experience in analytical chemistry, electroanalysis, medical diagnostic, and Dr. Warwick has very uh, good experience in from research to the biosensor development. So leading innovation in biosensor manufacturing, medical device development, and advanced biomaterial technology. And also Dr. Warwick has extensive efforts spanning biosensor design, electrochemical detection system, mass production engineering and regulatory strategy, and also with global market development. So please, Dr. Warwick, you can share the presentation. Time is yours. Thank you.
So, good morning, everyone. Many thanks for the introduction of my resume, and I would like to share the slide. So, okay. So everyone can see the slide.
Yes,
Yes,
yes.
it is visible. It is
Okay.
visible.
Okay, so today I think it's our pleasure to share some information to all of you. We are thinking about how to make a biosensor manufactured from design to mass production. I'm Warrick, and our company located in Taiwan. Yeah, so it's central of Taiwan is famous M.Si is like a Salmon Lake. So okay, we can go to the next page. That's my resume. My background comes from the research and development. So my first 10 years for the biosensor company, especially focused on the diabetes patient, the glucose sensor. So after that, we think about, I changed my career to move to the marketing and sales. Honestly speaking, if we just only think about the sales number is not good enough. So that's why we need to provide another value to our customer, not only the product performance, but also any possibility to improve the whole patient's usage and habit to improve their diabetes status better. So I spent around maybe 3 years for the marketing and sales, and then we established the company since the 2019. So my background comes from the analytical chemistry, especially focused on electron analysis. So I think previously the professor she mentioned a lot of things. I think we are at the same page. We can fully understand what is the limit and what is the points we need to overcome.
Sorry, Dr. Warwick.
Yeah.
I think your slide is didn't move. So maybe you can like slide
This
show
one
your.
is okay right
Yeah,
now.
it's not moving.
Okay, sorry. Okay, so
Let maybe
let
you
me
can
sorry, let me
like full screen.
yeah, okay. Yeah, it's okay.
Yeah.
Okay. Okay, so we go to next page. I would like to introduce my career. Okay, the first 10 years I served in the Bio9. I think it's a very good company because right now it's the top 3 biosensor company for diabetes patient in Taiwan. And each year we can have the annual revenue just only glucose meter and the strip make the revenue annual revenue around 2 billion, yeah, Taiwan dollars. So just only one product. But we also moved to the another field, not only for the diabetes, but also chronic disease management. And after that, I joined the Biosensor R&D Corporation. They provide the product for the basic research and the equipment. So we opened our business for the different territory like China, Singapore, Malaysia, India, Thailand, and Japan. And after that, we think about what kind of the value we can deliver to our customers. So we have our own company, we call it PICS. Originally, we provide the consulting service to help the researcher move cross the mass production more easier. It's like we can provide another kind of perspective to make them think about not only confirm the principle 0 to 1 is workable, but we need to think about okay, what is the next step to let you move more smoothly. So we provide a consulting service first, and after that, we also provide electrons like a screen print, electron sputter, or electron printing in the substrate. And we also provide a mass production technologies, means we can help our customer to build the factory in their location. So fully technology transfer from PICS. And HC agriculture material is for the agriculture purpose. So that's why there will be our another business. And during the 2023 and 2025, we also help the symmetric group, the subsidiaries in medical to restructuring and the transformation the medical business unit to make it better. It means to reorganize their product portfolio for the wearable device. So all about the printing and the chemical formulation. There's my career and okay, what can we do for the science and the biosensor field? Originally, we need to think about that. It's about a business. So, okay, it means not only research topic is good enough, but also need to export to the end user to let them think about, okay, it's worthy to make it, to have it. So, indispensable region demand from industry perspective is very important. We cannot judge the user for entertainment, lifestyle, or even social activity, but for the diet, like agriculture or the medical device, to saving lives, it must be choice. So that's why the PICS focus on to provide the material, the production technology, or even we can help our customer to make the equipment to ensure their topic can move to the mass production easier. So, okay. Who is the target audience of we want to have? Actually, biosensor can focus on the biotech, agricultural, medical device, even science approach. So most of the time, this company is very interesting. We don't have any website because most of our customers signed an NDA, non-disclosure agreement with us. It means actually we always in work back side of our customer provide the very key component of biosensor to them. So we provide a product and service to them. So not only biosensor substrate, but also medical device. Okay, so that's why it's not easy to find us. Normally, pass through the distributor or even the someone in this field can know us. And our location in Taichung City, and we have 2 sites, the office and the factory in the Science Park. So finally, I think after this meeting, if you have chance to visit us, we are welcome to let you know, okay, what is going on for the biosensor manufacturing. That's our milestone since the 2019 to 2026. Also, so okay, I think later we can share the slide to all of you. And we come from different, our members come from different learning skill and technology. So not R&D or quality control, even production art. The market service, market sales and service also, and we also help sometimes when the team want to have a new company like startups, we also help our customers think about how to make a roadmap of the company management like shares. So finally, this topic must be let some profit or outcome turn back to the members. I think it's more better to encourage the people want to move forward. Okay. So we have in headquarters, we have 4 members for the technology. Warrick, Dr. Ziv, and he focused on electron analysis and the principal confirmation. And Alex focused on the production and the printing. And Patrick Zhang, he focused on the equipment like a plasma surface pretreatment. And we also have a sales channel. Lucy, she focused on Taiwan sales territory. And we also have the overseas members in China, in Thailand. And we also have the consultant in Thailand. Like in Taiwan is Jeff. And we also have the US consultant from the Northeast University in Boston. So the professor, he do a lot of saliva testing very well. So I think we can have a lot of information to let all of you know if you need. So what is the expected value of what we want to deliver? Actually, our constitution come from different background and also have a very senior working experience, and we can compromise so many kind of field to provide the whole system service. So not the sale. Finally, okay, not only talking about the sales. Most of the time, our customer become our partners so long. So we provide a consumer electrons. We provide a production equipment. We also provide a consulting and the service possibility. So right now we have our facility, and we can show, okay, not only sales. Now actually we are manufacturer, and we can help our customer for the customization is very important because each kind of the design will be different. We have very precise equipment to make it, even the needle type, the electrons. So what does piece we deliver? Previously, the professor, she mentioned about, okay, we need to make some modification on the electrons, like, special chemical, like a polymer detergent, hydrophilic film as the stabilizer. And you need to put like enzyme, antibody, or aptamer, even DNA, nucleic acid as the probe. And then you can pass through the mediator to amplify the signal to let the electron receive that. So we start from the electrons, because if electron is not stable enough, and it's very hardly to get a low background and then make a SN ratio better. I mean signal to noise ratio better. So most of the time, our customer emphasize what is the problem, and they have an idea to solve it, and they can define why it's important, and we help them to test it and make a prototype to them. So I think not only, because most of our customers, they are so smart, they can have a lot of idea to solve this problem. We play a role to help them, make everything simple and not too complex. Okay. So we think about that. Most of the time, you can think about, optical, electrochemical, thermal, and the magnet as the signal to get the numbers and to measure it. And most importantly, for academic research, you confirm, what is the detection limit or how to prove the concept. But unfortunately, you need to think about the repeatability and the reproducibility. And most of the time, we are stuck on the reproducibility because the material, lot of variation, or even the production process too complex, too much. So that's why the stability is not good enough, especially if you modify too many layers on the electrons. Actually, the error rate will getting lower and lower because the performance will not be good. So that's why we help our customer try to reduce the steps, become the one step, not too much. Otherwise, the signal will become very poor. So our focus, we aim to 1 to 100. It means how to make the system integration and make the pipeline production go smoothly. Otherwise, it's very hardly to talk. Leonardo's, okay, we have the capability to make it real, not only in the lab. So the scalable batch production is very important for the medical, for agriculture, for environment, for the testing. It's very important. So that is we want to deliver this message and help our customer to do. So, okay, we know that. And what is the next step? It's not a fancy activity. It's really need to have the high reliable for the starting substrate. So you can think about if I want to have the high building, so my bathroom must be very stable and deep. And the mindset must be, okay, how to make it real stable. So we provide so many things, and we also provide some commercialized mindset to everyone. Most of the time, you need to think about that team is very important because of you come from research. Okay, the goodwill and the background is good. And you need to find the opportunity to win. It means you need to find a niche one to win, not the same like Leonardo's, because it's too competitive. But material is very important is because you need to find the material. Most of the time, we spend too much price or cost to make the material solve this problem. But unfortunately, when we move to the commercialized or mass production step, the cost and spending is too high. So that's why it's very hardly to convince the sales channel, make the business model or marketing strategy, and let the end user buy in. So that's why we need to go from the end user need. It means, okay, you need to have, okay, according to your topic, estimate the market share, estimate the pricing structure, and go back to think about, okay, how much real spending of this biosensor we can make it. Because normally we have the conflict between the academy and the real world because of the cost. So the next page is very important. The key to success to cost field survey, not research only. One, if RSD publish the papers, less than 5% is good enough. But normally for the production, we have too many steps. Okay, each step is we may have 95% good. And we have 4 steps. Finally, we just only have the 82% good to make it. So it's wasting too much. So that's why we try to help our customer to reduce the steps as possible as fewer and to make a high error rate. And right now, actually, in the lab, most of the time, we can make a research. It means, okay, prototype is okay, but the minimal viable product, MVP, I think the first generation for mass production, that is the focus we provide a service to our customer. And after that, okay, if you pass through this stage, you can commercialize this product. And if the production amount is higher enough, you can reduce the price, become the affordable one. So if someone say, okay, at the beginning, I want to have a lower price at the beginning, it's not real because the amount is too small. This page is very, very important. Before the mass production, please consider that regulatory, like ISO or IEC, precision, accuracy, operation environment, interference, and the long-term stability. Because, for example, like a long-term stability, we can think about, okay, open bio, the long-term stability, and the storage long-term stability. So it's about the quality. So it's very important. For example, like a diabetes patient, like a glucose meter, is need to follow the ISO 13485 and ISO 15197. So that's why it's very crude request from regulatory. And most importantly, okay, the market size, you need to finalize. It means okay, market size is means okay, what is the user prevalence and what is the user frequency? You need to calculate that, and it belongs to emergency or it belongs to the chronic request. It's very important also. And the major competitor, who is your major competitor? Is the top 5 over the world or territory who is the winner? So you need to identify, okay, when you want to have this topic to make the product in the end, you need to think about that. And the pricing structure is very important. Most of the time, honestly speaking, production fee will be less than 30% because the another cost was spent for the sales and marketing like 30%. R&D or administration occupy around 20%. So finally, even you minus the tax, just only leave the 20% as your profit is the optimized scenario. So that's why it's the real world we operate. So when we open this box to tell to the researcher, okay, if you just only think about the material price and the end user price, it's not fair. You need to think about the sales and marketing fee or even daily operation fee, not only production. So that is the real one and easier to convince the investor reasonable. Okay, so create a new business. Think about that. Okay, the biosensor, you need to choose the big one or smaller one. The big one is means that everyone do a lot of things and this market is very big, so everyone can occupy certain percentage. Is okay, like a diabetes. Or you can create a smaller one, make a niche one, create a demand according to you have very good result and your strategy is good and execution is very efficiency and you can win this one. So according to our thinking, we will help our customer to define, okay, are you in the innovator or challenger? Because if you go to the destroyer or replicator, it means it's not good enough. It's like a copy. So most of the time, 95% of new company and startups, they use the innovative technology, but for the all purpose, but performance can be improved. The market can be challenged. It means we think about cost-effective purpose. Yeah, that is in Asia. In Asia society, we focus on. But in Europe or in US, maybe they focus on, okay, 0 to 1 become the first one as a pioneer. Just normally, just less than 5% the startups can win and the survival. So if we think about the survival, the company survival, okay, or this topic can survive so long, most of the time it belongs to the market challenger. So a lot of things. Yeah, when you want to move to mass production, okay, stable product quality and client purchase acceptance, marketing visible and expansion, and team can get something back is the up of the iceberg. But below the iceberg, you have too many things you need to think about. You don't have enough time. You cannot speak very well, and the finance status always shortage, and you also fall into high pressure, and you need to encourage the team move forward, yeah, with some patience. So many things. I would like to use this picture and share all of you to think about if we want to from the research to commercialize, this one is very important to think about, okay, how many milestones or the points we need to overcome together. And biosensor actually is the radar to let you discover this world. If you understand previous slide, you can think about, oh, we can go for the biosensor application. So previously, like a professor, she mentioned, okay, you can modify something like basically you can have the bath electrons and make a surface modification. You can have the absorption, covalent bond, polymeritation, or even put some narrow material on it. What we deliver, we deliver the gold or carbon very stable base electrons or conductive substrate to you. So it means that if your substrate is stable enough, whatever you want to modify something on it, you will not find any system error or the electron error come from that. So it's more easier to let you solve this problem. Second, you can modify some kind of biomaterial like enzyme, antibody, DNA, aptamers as the probe, or even binding something for catalyst. So you can make some target can be analyzed. Or even you can make it to measure the current, the potential, the voltage. You can make the resistance. We call the current, potential, or impedance. What is the difference? If the difference getting higher, it means the concentration will become higher. So you can receive that. It's not the analog signal, it's the digital signal. So it's more easier to let you understand, and it's more easier to let you carry on. Because normally, if you think about the analog signal, everyone judge that it come from different concept. Even you see the later flow, 1 line and 2 line, and how deep of this color to make sure the concentration, no one can judge that precisely, but electron chemistry or electron analysis can do that because it's a digital signal. So who is the biggest marketing or very good successful example? Glucose meter. Actually, glucose meter has two kind of glucose meter. One is a portable one is like a traditional glucose meter in the strip, and the second is the continuously monitoring glucose system. We call it CGM. Or you can change the enzyme for the lactate or even lipid like cholesterol, triglyceride, and the high density or low density lipid. Yeah, it's okay. And this one is very good success story to all of you know, and actually the market size is pretty high. Right now, 1 in 10 who has a diabetes in the world. So second, what is the application we can approach? Small molecular, like a dopamine, like a catechol, like some kind of a small molecular weight, the species you can detect, or ions, like a heavy metal, like a lead, cadmium, potassium, sodium, or even the another ion make some pollution in the environment, or some biomarkers you can detect. And actually, use the redox activity to make that we call the cyclic voltammetry to see, okay, what is the real performance to set the voltage to detect this species. So that's why they can get a very fast real-time result and the low sample preparation. But I have to say, all about those one, they are very convenient, but everything relative how good of the chemical modification on your strip or the electrons. So the modification art is very important. It's very important. So not only make the electron stable, but also modify something on it. What is the species is very confidential or very important to make it stable, and make a selectivity is high enough. So if we want to go to advanced electrochemical biosensor integration, not only make sure, okay, it can see the signal, but also need to have the sensitivity specificity or the low limit of the detection is very important. And actually, right now, most of customers try to have one drop for 2 or 3, even 5 target can analyze at the same time. Actually, right now in our customer need, we already deliver the multi-channel screen print electron to them, like microfluidic. So that's why we also can make some kind of chip to integrate with their device. So It's very important. We have production technology, and we know what is the background you need, and we need to have a transparent communication. So most of the time, finally, our customer become our partner, or we become their partner because no any hiding information inside. So that's why we can open the data to exchange and the real-time feedback then, and make it very successful. So that's why we right now most of customer come from the disease diagnostic and the drug monitoring. Environment testing now is fewer because actually it's all about a policy. So if policy enforce, the testing must be the frequency must be higher. So I think the marketing will become good. But right now, I think most of the environment testing, they just try to get a sample, go back to the central lab. So if someone can make the sensor the accurate result almost or similar like the central lab equipment, I think will refresh the policy decision maker think about okay that the testing frequency getting higher. So we provide a core substrate for the biosensor. Simple is complex. Why? Because we provide different kind of screen print. We have a regular electron model. We have a factory. We can have a reasonable customization. For example, you can see the right side. For the palm sense, for the sensor, for the drop sense, each kind of the sensor design, we can make it, and even the performance better than theirs. And for the single electrons like 101 or 103 or 105, a different approach. Like 101 for the DNA nucleic acid, or even antibody or aptamer modification, because those biomaterial is so expensive, so you cannot drop and modify it too much. So that's why you need to think about how to reduce the cost at the beginning. 103 for the small molecular or enzyme. It means you drop enzyme modify on it is cost effective, is affordable one. The bigger one is for the small molecular, even the heavy metal detection. So it means just modify some polymer, low cost the polymer as the trap to make the heavy ion metal deposit on it, and you can measure it and stripping out. So different design, it has their own story, and if the attenders who has interest in that, and please let us know, we can share the information to all of you. And not right now, our carbon is not a pure carbon. Our carbon is the graphene, not the graphite, because graphene is more sensitive and more stable. So that's why now we use the graphene. And actually, we can also call it paper base, but actually it's the PP base. Because why we need to choose the PP? During the printing, we need to pass through the tunnel oven. The tunnel oven temperature up to the 120 degrees Celsius. So PC cannot pass through that. And the PET cannot pass through that. Teflon is too soft, but the PP is good enough because the PP can hold on 110 degrees Celsius without any shape change. So we choose the PP is the stable one, and we can make a single electron or 3 electron system like a working counter reference on the same surface. We have gold electrons for sputter. Yeah, we use a sputter. It means use very pure gold deposit on it and use the laser graving to make the shape. We also have another technology like electron printing. It means for the heavy metal use, and can make the cost a little bit lower. Yeah, not like a sputter. We also have the PT platinum working electrons, and normally our counter is the carbon. And our reference is the silver chloride ink. So that is our performance, and actually compare with the previously product, we can reduce the delta EP up to the 110 millivolt. maybe all of you may have enough the background to see, okay, how good is performance. But I just try to tell you, if you see this graph in the middle, this graph, if the peak more symmetry is good. If the peak is too separate, too much, I think the resistance will getting so high, so the background will become worse. So we also have some comparison from ours and Europe, the manufacturer, even Thailand, they make it. Use the same process to make the comparison and the testing by ferricyanide in the potassium chloride solution. Actually, right now, we can have the better one. More symmetry is more easier to get a low background and the ISD will be lower. So that's why the substrate is very important to all of you if you want to go to the biosensor field. And we also have the gold. So the same philosophy. And later on, I think I can share the slide because today we have so many information, try to share all of you. And we also provide equipment for lab industry scale. It means reliable is priority 1 to 100. It means the printing look like so easier, but actually you need to think of so many things like your ink, your tooling, your moving parts, or even what is the equipment, and the drying oven is very important, not the batch one, not the batch type. So many things we have to consider about that. And even the material, we need to select, okay, what is the plastic substrate? What is the ink? What is the tooling? Even the squeaky, even the pore size or roughness after drying. And the M.Si actually, right now we did not offer anymore, but if you need, we also open another page to provide this one. And the spacer hydrophilic fin also for the modification. That's our working station design. If you have chance to visit our factory, you can see all of them. Yeah, things from beginning from the material to the end, like a cutting. So print it, make it dry, use the plasma to make a surface pretreatment, use the dispenser coating the chemical on it, and the tapping on the hydrophilic fin for the capillary sampling, and the cutting to make a real one can be used. So I would like to use this picture to share this one. Okay, from left to right side, it means, okay, make an ink and print it out, make it dry, pass through the oven, use the plasma, make it hydrophilic performance and drop something on it, and finally use the cutting machine to make the shape. That is our equipment we can deliver, and we can help the customer to make the customization biosensor. And the plasma also very important because it is a key. After printing, we have a different kind of plasma to help our customer achieve a certain goal. So not only make a hydrophilic film, but also make some chemical functional group on the surface. So different material, what is the parameter, we can share the information to all of you. And originally, we have a very small space, and you can think about that just only 50 meters square. We then put everything inside, and we also let a lot of visitors to come to us to see what's going on. And most of our customers come from Japan and Thailand. And then right now, we already upgrade our factory. Now it's the second generation, almost everything automatically. So I would like to make a summary. I think that if you want to have mass production, dynamic, the tolerance window is very important. And the material must be stable. And most importantly, the design must be easier for the manufacturer. Because normally we try to solve the problem, use out our effort to solve that. But finally, it's very hard to produce it. And that's why to show some philosophy or some image to the granting agency or investor, think about, okay, this is good enough and we can move forward in the next step. So that's my summary to tell want to share all of you those information. I think it's a lot of information inside, but then after that, we will share this slide to all of you. So many thanks. Wish we can provide a good service to make your niche develop better in the near future in Indonesia. So many thanks.
Okay, thank you very much, Dr. Warrick. It's very interesting presentation about how the idea how to move from the research to mass production. I think is for manufacturer. There's many things to consider. There's many things to consider. There's many things to solve. And I hope in Indonesia there's like a lot of manufacture also for the biosensor development here. Thank you very much for your time and presentation.
Yeah,
and for the next one, we will have the last presentation from Mr. Jeff Chen. So Mr. Jeff Chen is Sales Director of Southeast Asia in BioDot. So Mr. Jeff Chen is a Sales Director of Southeast Asia in BioDot, and the BioDot is located in Irvine, USA, and he have extensive industrial experience in IVD market for over 5 years and familiar with biosensor and rapid test, R&D and production process. And he has joined the company since 2012 and used to be director of BioDot in China subsidiary. And he was worked as a sales director in sectors such as life science, pharmaceutical, healthcare, manufacturing, and nanotechnology. Before joining the company, Mr. Jeff Chen received his M.Si in degree from University of Georgia, USA, majored in immunology. So this time, Mr. Jeff will share to all of you about what kind of consumable, what kind of instrument that should be have in biosensor development and biosensor manufacture. So Jeff, time is yours. Please share your screen. Thank you.
Hello, everyone. So let me share my screen. Okay, so can everyone see my screen?
Still processing. Yes, we can.
Yes.
Okay, thank you. Okay, so this is Jeff Chen from BioDot. I'm responsible for APAC region as like Cariando introduced. So today I will try to introduce like what we can do for all the R&D researcher and manufacturer customers in biosensor and biochip field. Okay. So I'm not so sure like how many people here are familiar with BioDot, but I'm sure there are some. Okay. So this is the topic I will try to go through today. So who is BioDot? Application review, why BioDot? Solution offering, case study, and summary. Okay, so here is BioDot, okay, located in Irvine, California, and all the systems are manufactured in US. So let's say sometimes if you hear people told you like this system of BioDot like produced outside Irvine, US, that's not true, okay. Everything of the BioDot system are manufactured in US. Then this is our like lovely office and the manufacturer with all kind of like the patent, everything there. Okay, so BioDot has been on the market for a long time, like since 1996. And then we focus on later flow, some of you, the audience here knows. Then we also focus on biosensor, biochip. Then we do a lot of like solution for the customers, especially for manufacturer. So right now, all the manufacturing is in Irvine, California. Then we have sales and service office in Chichester, UK, and Shanghai office in China. Then we have distributor in apex from Japan, Korea, Taiwan, Thailand, Vietnam, Singapore, Malaysia, Indonesia, India. Then so far we have like more than, I think 2,000, actually close to 2,500 customers. Then sell over like 60 plus countries. Then with, of course, we have a lot of patents. Then BioDot has been acquired by ATS Automation back in 2021, finished by 2022. So right now we are like a subsidiary of ATS Automation. ATS Automation is a big company. It's like listed in Canada, like a stock market, and also in Nasdaq. Then the overall revenue for our mother company, of course, is quite big. Okay, it's over 2.5 billion in average per year, and then we cover life science, food, beverage industry, consumer packing, also in energy. So it's like after we join ATS big family, we also have the larger capacity and capability to help our customers from, I mean, almost every angle. Okay, so this is our focus today to discuss. Okay, so BioDot's product line covers cytogenetic, medical device, then point of care and industry pharmaceutical related. Then, so you can see we have customer covering almost like life science field, and some are in pharma, and most in medical device. So today I'll try to focus on biochip and biosensor. So this is our like kind of like a slogan. Okay, we are creating a world where every scientist's vision is met with the precision it delivers. So just like the previous two doctors introduced, okay, in biosensor, biochip, actually precise is quite important, not just measuring. Then also when you deposit the enzyme or reagent, it's quite important. And then that's what we focused. So for BioDot, we have all kind of dispensing technology depending on what you are doing. So through the presentation, I'll try to explain one by one. But basically, we have like a microliter line and coating dispenser. We have nanoliter to microliter drug dispenser, and we have like a dispenser down to picoliter. But to be a lot of time, I will tell the customer, let's say, if you can make your test bigger, then try not to go small. Because like small is quite difficult. Because let's say once you go down to picoliter, you will see a lot of things changed. So it's like I always encourage customers when you do the R&D, try to make your things, or your design in the beginning larger, and then try to shrink it like gradually. It'll be better for your work, and then easier to complete your work to get a result. So how big is small? So that's a very interesting question, and then a lot of people are not aware of it. So just to give you some idea, okay, so human hair is around, the diameter is around 70 micron. Then deep your naked eye, okay, it's about 40 micron. But because we see this all the time, right? So maybe you can see like a 30 micron, like a larger like droplet. But to be honest, you evaporate very quick, okay? Especially you are in the like a low humidity environment, or the environment is very hot. Then the white blood cell is around 25 micron, red blood cell is around 8 micron. Okay. So then this ring here, just give you some idea. So for example, like a microarray, let's say in a 1x3-inch glass slide, you deposit 2,000 drop on it. Normally the drop is around like 200 micron. Then it means from this chart you can see, right? It's like around 20 nanoliter or less. Then as you can see, up to 250 nanoliter, the diameter of the droplet could be around like 1,000 micrometer. So just give you an idea by this chart. So when you try to design biosensor, biochip, even microfluidics, it's a good reference. But of course, this is not like a absolute number, right? This is depending on your surface treatment and then also the surface energy of your substrate, it will vary. Then so that's something you need to like pay attention in the beginning when you do R&D. Okay, application review. So biochip, including devices such as microfluidics and microarray, they used to perform many tests at the same time. For example, gene chips, okay, that's like developed since 1999, still used, but it's very useful to analyze thousands of genes simultaneously. But right now, this kind of application most of the time is used for gene screening and then some pathway finding, not really used for diagnostic. Okay, then biosensor, just like previous two doctors said, okay, is quite widely used. But of course, the most like important is glucose monitor, and then very popular. Then of course you begin to see like some urine test and some other tests become more and more popular. Very simple and then easy to use and easy to get a sample, either from your urine or from your blood. Then the traditional string printing process for biosensors are unsuitable for most of biological materials, particularly for the high temperature used to cure the printed electrode. So the alternative is to dispense the material. That's like something Dr. Warrick just introduced. So that's also something you need to pay attention when you try to make biosensor. Then here is kind of like a application overview, and then for biosensor and biochip. So biochip, you prepare reagent, print biochip, add sample, label, hybridization. Then you get a result, right? Then biosensor, similar. Prepare reagent, print, dry sensor, syndicate, assemble. Okay, so what BioDot does actually here, printing biochip, assemble, and in cytogenetic, we can also help to do hybridization and wash. But most of the time, we focus on these two, print, and then like add sample. Because that's how we do, precise dispensing on the precise target area. So why BioDot? Okay, because like actually on the market you can see some similar like a dispenser. Okay, but most of the customer or R&D researcher will face like a change volume, drop volume with low coefficient of variation, which is called like a CV. Okay. Then you want to adjust chemistry, then often leading to change in physical property. The most important, viscosity. Then adjusting drop spacing, then applying multiple analyte. BioDot can do this, and then we have been doing this for a long time. So, for example, like in this picture, this is biosensor, then this is biosensor, then this is a multiplex PC and like ELISA. Then this is kind of biosensor too. Then in order to fit in different applications, you find a proper tool is important. And then of course, the key, CV, precision. Then what's this like precision and mean? Okay, so volume, picoliter to microliter as a single droplet, viscosity 1 to 100 cp. Then less than 5% CV at our lowest volume, 25 picoliter. So for example, in like blood glucose sensor, our like this dispensing volume for the reagent normally is around 0.5 to 1 microliter. We basically can achieve within 2% CV. That's what we do. And then 2% CV, what I mean is like, let's say you dispense like 1 million drop per day. by this kind of automation system. Okay. Then if you are dispensing like a more like a water-like solution, or I say like a buffer, you can easily achieve 1% CV below. Then the motion precision normally is around less than 10 micron. But like if in reality, when you do production, right, I would say below 25 micron. But 10 micron is the limit of the mechanical design. Then speed, I will show you. It will be very fast. Non-contact on the fly dispensing. That's designed for manufacturer, but of course for R&D, you don't need such like a high speed, right? You can choose like different mode to do that. Then substrate versatility, biosensor sheet, wafer, PCBs, microfluidics, and custom novel designs. Then in biosensor biochip, just like Dr. Su just mentioned, okay, the plastic is very common, right? Then we also have customer use wafer to do the biochip. Even like a TSMC right now, everyone is familiar, right? We have been doing something for them. It's like use like an 8-inch wafer to do some biochip, and then microfluidic, of course, depending on the design. Then flexible channel configuration. For R&D, most of the time you just need one channel, but for production, you can see like this, okay? More than 10, 12, 16 or more, depending on the manufacturer requirement. We can always like customize that. Then enabling technologies, process QC, okay? For manufacturer, the most important is stability and QC. Even for R&D the same. Like how can you ensure what you are doing is correct, especially the dispensing amount? So for different kind of system setup, we can have like a horizontal camera, vertical camera to check the position and dispensing volume. The camera can capture the dispensing droplet size, then do some internal calculation to match the volume you desire to dispense. Then
Terus
you
gitu kan
can
nanti tambah lagi 50 jadi total 100.
Oh iya,
Berarti
mau
mau
langsung 100.
kalau mau istilahnya ada sisanya
Oh,
diturunkan
kayak
lagi
mencuci ulang ya.
Oke. Siapa tahu sekali tadi belum turun semua.
Berarti 50 ya.
Sorry, any questions?
Sorry, Jeff. You can. Yeah, I
Continue?
already mute. Yeah, you can continue. Yeah.
Oh, okay.
Sorry.
Don't worry. Okay. So then you can add like a cheer sauce or like a target location. Cheer sauce, like preparation is sometimes quite important. Let's say when you're dealing with some reagent, right, especially antibody or protein, you like temperature to hold it. Because for the whole process, the production of R&D process, you may need like a few hours, right? Then you need to your antibodies protein to be stabilized. So your temperature may be around 20 degree Celsius or below. Then chill sauce sometimes very useful. Then humidity control, as I just mentioned, okay, so when you try to dispense like small volume, so for example, you are dispensing 100 nanoliter, then if you like environment humidity is below, let's say 50, you may see the droplet evaporate very soon. So you need to control your humidity above some level. Normally it will be around 70% or higher. It will be better for your like production or R&D like result. So for example, you try to dispense something, and then today sunny day, humidity 60, temperature 30, then you dispense very well. But tomorrow, like humidity is very high, 70%, and then your like temperature drop down inside. Then you may see your dispensing result or like your test result will vary. So that's something very important. You need to always control your like environment, humidity and temperature, all the same. That would be better for you like a test. Then system enclosure, if necessary, you can incorporate that. Then process optimization. So on board high efficiency tip washing, because like let's say we dispense like a high concentration of like protein antibody, let's say above 1 milligram per mil, up to 5 milligram per mil, then your protein may try to like crystallized outside of the dispenser. Then what do? You cut, right? So you need to wash it from time to time. But how long to wash one time? The wash cycle, you need to try then to understand, because that will be depending on your like buffer system, your formulation, and then your concentration. Then software for highly complex process. For this part, we can customize the process to match your production process. But for R&D, you be just like to mimic or like make your operation all the same at one, like as you do per day. So depending on the requirement again. Then of course we have some other options. So heated and chill fluid paste. For some reagent, we need to heat it. Then in order for it to flow. So for example, sometimes we will dispense like wax, and then, or for example, like some the qPCR cover. There's something it's kind of like a different like material, but when we try to dispense that, we need to heat up the wax or like some other material, then we can dispense it. Then temperature control, shaking mixer of source solution, material handling, real-time good and bad part inspection, upstream, downstream of automation integration. As I say, ATS is very strong and experienced on automation, so that's something we can do too. Okay, so this is some example staking. This is just like a process, okay? So you can have like a loading the material, then dispense, then goes out, then goes dry tunnel. That's kind of like a setup for the biosensor. So for biosensor and biochip project, okay, especially for the manufacturer, we will need to like learn what the customers like project and also the goal they want to achieve. Then of course we need to understand every like a single detail process, so we can help to design the system to match their like project. But for R&D, I always try to ask our customer what do they try to achieve. Then of course by our like experience we can really help a lot. So that's like what we do to help to the customer. Then this is also an example. Let's say this is a sensor. And so, but this sensor, we need to deposit on this like small dot here. So this part, we try to deposit on this like a small area. Then like we need to design, and then it's like reagent has some particle inside. So we need to design like a shaker and also chiller to help the reagent kind of like always stirred, and then those like material inside will not precipitate. This is like just another example. Sorry. Okay, so the diamond range and precision. Okay, so BioJet and Rainmaker. So BioJet is the most common used dispenser type for biosensor, and Rainmaker is like for picoliter. So as you can see on the future here, so the volume range for BioJet is around 2 nanoliter to 2 microliter per droplet. So it's step repeat and on the fly. Precision, normally we will say less than 5% CV, but depending on the formulation, we can easily achieve like below 2%, even 1%, depending on what you are doing. Then for Rainmaker, this one, I think for the past few years, the most common use is for continuous glucose manufacture. That's what we do a lot. Okay, so it dispenses from 25 picoliter up to multiple milliliter. But for single droplet, I would say from 25 up to like 200 picoliter. Again, it's step repeat and on fly. I'll show you in the next video. Then normally it's less than 3% CV. So here is some videos you can take. So as you can see, inside, okay, we can dispense a multiplex like ELISA, right? This machine can easily dispense a 4x4, 16 droplet inside. Okay. Then the next one, this is Rainmaker. Then this one we call like random like a dispense. Okay. It means like each tip, there are two tips here, right? You can program it to dispense like in different sequence. It's not necessary one by one. It's like you can dispense like a different tips in different time, not by sequence. You can design it. The next one is on the fly. The most common use for the manufacturer. So as we can see, try to do right, it can dispense by different mode. So you see the motion precision speed, okay. How long does it take to dispense 5,000 unique chemistry to 150 device with 12 dispenser channels? So step and repeat. Normally we can see 215 hours. Then on the fly, like 3 days. Then random access on the type fly, 15 hours, depending on how you want. Then I can give some small idea. Okay, they say for the blood glucose sensor, they say normally we produce, they say one sheet is 100 sensors, right? Then we can easily produce 1,000 up to 2,000 sheet per day by 2 channels. That's all we can do. So you see 500 by 1,000 is how much? Half million? Easily. Then 1 million you can achieve in a day is very easy. But For R&D, you don't need that kind of like speed. What you need is precise in the beginning. Okay, so substrate versatile platform option. Okay, so for the most of R&D, okay, you don't need like a big machine. What you need is like a benchtop system. So this one is benchtop, 80-15-20. Then if you need like let's say if you need picoliter and microliter dispenser at once, then you can choose Omnia. Then the size, okay, for 80-15-20, you can have like a different like setup for we call nest. The place you host your material, okay. So it can be 14 slide holder, it can be 4 microliter plate, or like totally plate. We call magnetic and hold down like a nest. The same in Omnia. The size is larger, okay, 27 slide, and you can be totally flat, or you can put microliter plate. Okay, then dispenser range here, 2 nanoliter to microliter is for 80. For Omnia, picoliter, 25 to microliter. Then position verification, vertical camera. Group type, compound, DNA, protein, cells, okay. Labware option, plate, slide, micro device, sensor, custom. Then some basic price, okay. So depending on what you do, you can choose from different model. But for R&D, then you can choose this one. That's easier for most of R&D. Then flexible channel configuration. So come back here, okay. So for 80-15-20, okay, the maximum you can have 4 channel, and then that can be easy upgrade in the field. Means like for R&D, you can just need to have 1 channel in the beginning, then gradually upgrade to 4 channel. Only the same, up to 4. Then this is like some like options R&D we never use. Okay, so for example, like this we call a head theta rotation. Means like when you try to dispense the sheet, but sometimes the printing may not be 100% 90 degree. So we can try to use a vertical camera to check the shifting angle and then guide this like a theta rotation head to match the angle, then to dispense. Okay, so we can do custom orientation spacing and then different dispenser type. Then the machine you see here is we call 86,000. This is designed for large manufacture, mass production. Then QC process, okay. So this is for picoliter dispenser. Simply, because picoliter is too small, you cannot measure it. So you need to use like a camera to capture the droplet, then try to do some internal calculation to see what kind of size you are dispensing. For nanoliter or microliter, it's easy, okay? We can use like Empendorf, then to accumulate, to collect the droplet, and then they say you can accumulate, say, 100 drop, then weight on the balance. So it's easy for you. But picoliter will be different. There is no, it's too difficult to measure it by balance. So you need to do a different way. So high frequency imaging, and then strobe upscope, lightning to capture and measure individual droplet. So we will see the morphology, volume, and then do statistic calculate to check the CV. Then high magnification vertical camera for device imaging. This one in 80-15-20, you can see this too. So before and after, you can try to take image and check where you dispense and then where is the location. So droplet position correction, target alignment. And then the software can also make it automatic. We call fiducial finding. So for example, let's say you have a device, then you can set a target. Then the machine, the camera can find a target to dispense. We call it fiducial finding. It's very useful for some microfluidics device to dispense. Then as you can see, the offset, for the targeting is quite precise, okay? Normally less than 10 micron if you use the fiducial finding function. Then option, okay? We can adapt to various levelware. Again, depending on what you do. But as you can see here, okay, this one is for slide, right? Then this is for microtiter plate. The most common for biosensor biochip is this one. Vacuum and magnetic nest hold the slide or material on the surface. Various workable options allow for placement for plates, slides, trays, custom device. Then we can customize shaker, temperature control for source, destination, plate, and tube. Okay, so it's near the end, okay? So that's BioDot journey, okay? So we develop on scalable technology platform, meaning, let's say you use no matter which platform you use, you can use almost the same parameter in this product line. So for example, if you use AD1520 to do the R&D, then you succeed. Then you can transfer this parameter to AD6020, even the larger one we call modular, is fully automation, this kind of system. So it's kind of streamlined to help you work, from R&D to manufacturer. Then this is a ship-based biosensor production system I just introduced before. So inline batch, again, use AD1520 to practice, then migrate to AD620, even larger. So for this one, you can see this is a kind of magazine, okay? It can upload the material onto a dispenser system automatically. Of course, this by customization. Everyone's sheet will be different. Then for the past few years, I should say for the past as you can see, 2009, we have many publications on the website, so feel free to find it. And then if any questions, come back to me. And then summary. BioDot has necessary tools to manufacture biochip and biosensor, of course, from R&D status. Then our wide dynamic range is used to dispense a wide range of reagent. Improve CV for current assays is the most important. So less than 1 CV and volume above 1 microliter, that's very easy for us. Reduce volume and cost per assay. That's our goal to help our customer doing manufacturing. Increase density of assay. Of course, that's important too. Then because it can cost down the manufacturer. Then we have experience manufacture DNA, protein, polymer, and cell-based biochip. Then our tools for picoliter and nanoliter droplet placement are critical for high density, low volume printing application, including biochip and biosensors. This is also, I forget to mention one thing, okay, so for nanoliter droplet, okay, right now it's also quite useful for NGS sample prep, especially you try to build up the library. We have customer use this for sample prep, Takara, and then some other third-party laboratory in US. So let's say if you have a NGS application and you need our help, you can approach me too, okay? I can share some experience. Okay, thank you. That's my presentation today.
Okay, thank you very much, Jeff, for your very insightful presentation about instrumentation for manufacture of biosensor from R&D to production to mass production. I think. So, and then now we enter the Q&A session. I think this webinar will be so long because we see there's so many questions in the chat room, Prof. Yeni, Dr. Warrick, and Jeff. So I will start to read one by one. the question. Okay. I will start some question from the chat. First, for Prof Yeni ya. Prof Yeni, untuk Prof Yeni mungkin ada beberapa pertanyaan Prof yang peserta ini banyak sekali yang tertarik dengan teknologi biosensor ya. Jadi ada beberapa pertanyaan mungkin bisa dijawab beberapa, nanti juga kita akan lanjutkan dengan diskusi kalau misalnya masih ada pertanyaan. Mungkin yang pertanyaan pertama adalah untuk pembuatan biosensor sendiri, misalkan ada enzim yang bisa digabungkan dengan matriks seperti grafen atau yang lainnya. Apakah ada pertimbangan dalam memilih matriksnya? Kemudian metode apa yang bisa digunakan untuk penggabungan bahan biologi dan juga matriksnya. Jadi pemilihan metode pemilihan matriksnya, kemudian bagaimana menggabungkan antara bahan biologi dan juga matriksnya. Mungkin itu dulu, Prof. Nanti kemudian dilanjutkan untuk pertanyaan selanjutnya.
Oh ya, baik, terima kasih atas pertanyaannya ya. Misalkan enzim bisa digabungkan dengan matriks nanomaterial gitu ya, atau grafen atau lainnya. Itu tergantung di tujuannya ya, tergantung elektroda yang kita gunakan. Misalnya kalau uh, itu berikutnya kan ada yang bertanya pakai pasta karbon. Nah, kalau elektrodanya pasta karbon, yaitu menggabungkan dengan matriksnya bisa di bulk modification gitu ya, jadi bulk modification, jadi dicampur gitu langsung ya. Tetapi kalau misalnya elektrodenya SPE, eh, apa namanya itu di broadcasting di atas permukaan elektroda, jadi hanya di permukaan saja. Tetapi apakah enzimnya itu bisa digabung dengan grafennya, misalnya di sini ya, kemudian di dropcast di SPE, atau kalau tadi kalau dengan apa pasta karbon bisa digabung langsung gitu. Nah, itu kalau kita melihat biomolekul sifatnya, supaya dia itu bisa terimobilisasi di permukaan elektroda itu ada macam-macam tekniknya, ada misalnya adsorpsi saja. Jadi kalau absorpsi saja tidak digabung dengan materialnya. Kemudian bisa juga di apa namanya itu, di entrapment gitu ya, dijebak dengan suatu matriks membran biasanya, karena kalau enzim dia itu eh, karena memiliki aktivitas biokatalisis gitu ya. Ketika dicampur itu atau digabung tadi, apakah akan hilang aktivitasnya? Karena dia ada yang harus dijaga aktivitasnya yang ditentukan oleh strukturnya. Jadi strukturnya tidak boleh rusak, kemudian juga dia harus terlindungi. Sehingga ketika dicampurkan dengan suatu matriks itu harus yang memang tidak merusak enzimnya gitu, kalau ini enzim, atau sama juga ya antibodi dan lainnya, karena secara struktur tidak boleh merubah strukturnya. Kalau dia dijebak saja, entrapment saja dia bisa, atau di selain dijebak, di enkapsulasi gitu ya, di si enzimnya itu semacam dilindungi dalam suatu kapsul. Kemudian ketika dia ada substrat atau ada target, dia akan keluar dari kapsul itu. Nah, kalau misalnya tadi si grafen tadi bisa membentuk seperti kapsul, atau dia menjebak tetapi tidak merusak strukturnya, itu bisa saja. Jadi harus harus dipelajari eh, dari segi kematchingan strukturnya gitu ya. Itu tekniknya berdasarkan apa gitu ya, apakah bisa dicampur atau tidak. Nah, umumnya enzim itu kalau untuk dimodifikasikan ke elektroda tadi, supaya dia tidak, karena tantangannya tadi ya, dia akan mudah rusak dengan pH, suhu, sehingga memang harus ada pelindungnya. Tapi apakah pelindungnya itu strukturnya yang seperti apa gitu. Mungkin gitu ya. Apakah metode apa yang bisa digunakan untuk penggabungan bahan biologi dan matriksnya? Ya seperti tadi ya, metodenya enkapsulasi, entrapment, atau crosslink gitu ya. Ada senyawa-senyawa yang dia membentuk silang-silang sehingga enzimnya membentuk silang tapi tanpa mengurangi aktivitasnya dan mengubah strukturnya. Mungkin gitu ya, Pak
Ya,
Pak
baik
Rudi.
Prof, terima kasih. Kemudian ini ada juga pertanyaan mengenai ada salah satu peserta kita ini sudah menggunakan biosensor, Prof. Namun ternyata ketika di lapangan itu kadang-kadang dia alatnya itu tidak mampu mendeteksi yang terlalu rendah dan juga terlalu tinggi sehingga eh, dari pertanyaan tersebut beliau bertanya bagaimanakah ataupun bagaimana caranya agar si biosensor ini bisa mendeteksi target yang jumlahnya sangat kecil, misalnya dari nanoliter sampai dengan mikroliter seperti itu.
Oh iya. Tapi yang ini tadi belum dijawab semua, bentar.
Oh,
Yang
yang mana?
eh, yang ini elektroda karbon dengan SPE, apakah biosensor bisa digunakan? Ya, jawabannya ya pasti bisa ya. Mau pakai SPE atau pasta karbon, dua-duanya bisa, tapi pasta karbon umumnya bisanya untuk riset gitu ya. Kalau untuk POC, tadi POCT kurang efektif kalau pasta karbon karena dia tidak renewable, kekurangannya jadi kurang homogen gitu. Tapi kelebihannya tadi bisa dicampur dengan bioreseptornya. Nah, kalau SPE kekurangannya ya dia tidak bisa dicampur dengan bioreseptornya, tetapi SPE lebih match ke misalnya ke smartphone gitu atau ke device yang lain yang untuk portable gitu. Jadi semuanya ada kekurangan-kelebihan, tapi untuk penggunaan biosensor bisa dua-duanya. Kemudian juga teknik penambahan modifier tadi book atau dropcast atau spray, teknik mana yang paling baik. Jadi secara absolut tidak ada yang paling baik yang mana. Tadi tergantung aplikasinya untuk apa ya. Jadi kalau tergantung elektrodanya juga. Tadi kalau pasta bisa bulk atau bulk modification ya, bulk modification. Kalau SPE tidak bisa, bisanya surface modification. Nah, kalau drop casting itu ditetes, kemudian di apa namanya tuh spray itu disemprot ya. Nah, itu kalau bulk itu punya kelebihan distribusinya lebih homogen, lebih stabil gitu ya. Transfer elektron bisa meningkat dari seluruh volume. Tapi kekurangannya yang bulk itu konsumsi material jadi lebih banyak. Kemudian kontrol permukaan juga jadi kurang spesifik. Kemudian kalau drop casting, kelebihannya sederhana dan cepat, tinggal tetes gitu ya. Tidak butuh alat khusus. Cocok hampir semua elektroda kalau drop casting. Tapi kekurangannya distribusi biasanya tidak terlalu homogen ya. Reproducibility-nya menjadi rendah. Jadi di setiap bagian permukaan elektroda itu bisa jadi tidak sama. Kemudian film pelapisnya juga bisa jadi tidak stabil itu ya, ada keretakan misalnya. Kemudian kalau spray coating itu bisa lebih homogen dibanding drop. Di setiap bagian permukaan elektroda bisa merata gitu ya, dan lebih scalable menuju produksi, tapi butuh alat khusus. Dan ketebalannya apakah bisa dikontrol? Yang bisa ya kalau yang spray coating. Jadi ada optimasi parameter yang lebih kompleks gitu ya. Kemudian ini bagaimana membuat biosensor yang mendeteksi dari nano hingga mikro. Nah, itu optimasi suatu biosensor itu sangat penting gitu ya. Tadi harus ditentukan rentang linearnya di mana. Pokoknya harus linear dulu supaya tadi, nah pertanyaan berikutnya yang tadi disampaikan Pak Moderator, kalau jadi terlalu tinggi atau terlalu rendah, nah itu berarti perlu validasi, perlu dioptimasi berapa LOD dari si peralatan biosensor itu yang kita kembangkan, dan berapa linear eh, range-nya ya atau rentang linearnya, apakah nanoliter atau mikroliter. Nah, biasanya kalau pengujian di riset itu diuji yang misalnya rentang, misalnya keberadaan suatu biomarker rentangnya nano, ya kita uji dulu di level nano. Kalau misalnya tidak terdeteksi atau terlalu udah ditingkatkan pun nanonya tidak berubah, makanya itu baru dirubah mikro. Itu tergantung targetnya apa, dan tergantung luas elektroda yang kita gunakan, juga tergantung material elektroda yang digunakan. Eh, untuk memiliki rentang yang tinggi dari nano ke mikro misalnya, itu harus perlu optimasi atau perlu pengujian gitu ya. Mungkin gitu, eh, Pak Moderator. Nah, yang selanjutnya tadi apa tuh?
Yang selanjutnya itu tadi ada pertanyaan mengenai lanjutan dari pertanyaan tadi, Prof. Itu adalah apakah ada eh, biosensor untuk mendeteksi bakteri patogen pada jumlah sampel kurang dari 400 mikroliter, seperti Staphylococcus misalnya.
Oh, eh, kalau yang saya tahu ya ada gitu ya karena apa namanya itu mungkin sudah banyak tersedia dan visible ya. Eh, karena prinsipnya kalau untuk mendeteksi bakteri bisa metodenya banyak gitu ya. Bisa imunosensor pakai antibody antibakterinya, bisa aptasensor pakai aptamer, bisa DNA-nya, bisa juga AIS gitu. Nah, dan volume yang kecil biasanya kalau yang di POCT yang udah portable gitu memang volumenya kecil dan eh, kalau tadi konsentrasinya di bawah 400 gitu ya, di bawah 400 ya ya tadi tergantung LOD dari si biosensor yang dikembangkan itu. Jadi perlu perlu pengujian ya kalau riset. Tapi kalau misalnya udah yang komersial, kita bisa lihat itu LOD-nya berapa gitu. Atau LOQ-nya ya, karena yang terukur biasanya LOQ. Itu berapa karena apakah bisa di bawah tadi ya, di bawah 400 volume ya ini. Volume di bawah 400 mikroliter. Kalau alat-alat PO City yang diteteskan itu bahkan lebih rendah dari 400 mikroliter itu tadi bisa 20 mikroliter saja. Kalau volume ya. Ini ditanya volume atau konsentrasi?
Kalau di situ tulisannya volume ya, tapi sepertinya lebih ke arah mungkin
Konsentrasi ya. Kalau
iya
volume
betul.
ya kalau POCT biasanya memang kecil volumenya. Ya, 10,
Hmm.
10 mikroliter aja bisa gitu diteteskan ke
Mungkin lebih ke sensitivitas, Prof. Seberapa mampu mendeteksi jumlah bakteri yang sangat sedikit seperti itu, biosensor itu.
itu. Kalau yang berbasis DNA tadi ya DNA apalagi yang CRISPR tidak perlu amplifikasi itu kan bisa sangat kecil bisa langsung terdeteksi bahkan di bawah ya selevel PCR gitu ya batas deteksinya sangat rendah.
Hmm.
Eh, tetapi kalau misalnya berbasis DNA itu karena DNA itu adanya di dalam sel harus kita perhatikan metode untuk lisisnya gitu ya untuk mengeluarkan si targetnya dari dalam sel gitu. Tapi untuk performa optimal biosensor di klinis ya fokus utamanya ya tadi desain sensornya, validasi dan kontrol interferensi gitu. Seperti tadi hormon bisa lebih rendah atau lebih tinggi ya bisa jadi karena interferensi dari di dalam matriks gitu ya. Ada hormon lain yang juga dia mengganggu pengukuran. Jadi itu harus divalidasi gitu ya, pada intinya, dioptimasi, validasi gitu. Kalau untuk
Baik.
riset.
Terima kasih, Prof, atas jawabannya. Mungkin ada satu lagi terakhir ini. Eh, Pertama adalah mengapa biosensor berbasis POCT ini belum sepenuhnya menggantikan uji lab konvensional meskipun menawarkan kecepatan dan juga kemudahan gitu. Apakah apa batas fundamental yang mungkin masih menghalangi kok dia masih belum bisa dijadikan sebagai mungkin pengganti ya
Iya,
uji berbasis lab gitu.
ya karena seperti tadi disampaikan ya bahwa tantangannya itu apa namanya tuh reproducibility gitu ya. Kadang si materialnya itu tidak tidak konsisten. Kalau misalnya tadi ya kalau dari skala produksi itu juga ketidakkonsistenan material, kemudian juga eh, kemudah rusakan dari bioreseptor, yaitu tantangan-tantangannya. Jadi bisa berubah-ubah. Kalau kita sebetulnya kalau kita mengecek apa kadar glukosa darah gitu ya di misalnya di apotek A, apotek B, apotek C dengan merek yang berbeda bisa jadi beda-beda. Tetapi itu untuk biasanya perlu untuk apa ya diagnosis awal gitu ya atau diagnosis dini sehingga untuk memastikannya tetap perlu pengujian yang lanjutannya gitu. Tetapi untuk monitoring gitu ya di dini gitu itu lebih banyak digunakan yang POCT biosensor ini. Pertanyaannya mengapa apa belum bisa menggantikan secara sepenuhnya? Ya karena ini kan dibuat tadi portable seminimal mungkin, secepat mungkin. Jadi tidak sesempurna instrumen yang lebih kompleks gitu ya, kemudian apa metode deteksinya yang lebih apa ya, lebih mendalam gitu ya. Jadi memang penggunaannya tuh bukan misalnya untuk menegakkan diagnostik ya. Seseorang misalnya mengecek apakah ini tumornya ganas atau tidak, misalnya kemudian dicek sel kansernya. Nah, kalau misalnya pakai POCT ya tadi ya untuk dini, oh ini perkiraannya segini gitu. Tapi untuk pastinya mungkin ada cek yang lebih kompleks tadi ya, misalnya dengan CT scan, MRI gitu ya dan lain-lain gitu dengan instrumen yang lebih besar. Tapi tadi kalau untuk di daerah untuk apa? Ya tadi untuk dini gitu paling gitu. Karena diminiaturisasi gitu ya, jadi mungkin dibikin portable jadi tidak se apa ya, tidak se ya bisa berubah-ubah gitu asal tidak sekonsisten yang itu, yang instrumen besar. Seperti itu. Makanya itu challenge gitu, itu tantangan untuk stabil.
Iya, baik. Terima kasih Prof. Yeni atas jawaban-jawabannya. Mungkin nanti untuk para peserta yang mungkin ingin nanti menyampaikan pertanyaan, mungkin bisa disampaikan nanti di kolom pada saat registrasi pertanyaannya. Mungkin nanti akan kami sampaikan juga ke Prof. Yeni kalau misalnya masih ada pertanyaan tambahan. Seperti itu, Prof. Yeni. Mungkin kita akan beralih ke pertanyaan selanjutnya, to the next session for question from for Dr. Warrick. So when we discuss about move from research to mass production, approximately how long does it take to like biosensor development from research to like manufacture, Dr. Warrick? So
Okay.
maybe
Yeah.
how long did the process from like, and we have like, we already have the receive, and then how long to be like mass production product that can be produced as a mass production
Okay.
in
So
your experience?
Okay, according to the research topic, if you confirm the principle and make is the prototype, and we will pass through we call the VMV principle, verify the validation principle. So it means okay, for example, you need to produce it in the lab for the 3 lots. Small amount and just only maybe 100 or 200 pieces for each lot. And we will send to the third party to verify, okay, can be repeat or not. If it's possible, it can be repeat, and you will pass through around maybe 3 months because they also test the long-term stability or isolation test. It means, okay, they will put into extreme condition like high moisture or high temperature to check the performance is good or not. Unfortunately, most of time for the lab, the condition is very comfortable. So it's just available for the 20 to 30 degrees Celsius because of air condition. So they will inform you, okay, can you have any like correlation? It means, okay, low temperature, you need to multiply certain factor to back to the normal room temperature result, or even a high temperature or high moisture, you need to multiply or divide a certain factor back to the normal condition. So the first stage will take around 3 months, and after that, they will use their X6 production line to help you produce it, not by your hand, by their hand. Yeah. For example, we are the one of a third party. We can help to produce it by our hand. It means that nobody can touch that. I mean, the original member, to make sure it can be repeated or reproduced. And then, okay, check again if it's possible. And during the moment need to take around maybe 9 months. So it means if you commercialize before you want to apply or submit a document for regulatory agent, and you need to provide the first 3 lots from lab for verify the performance is okay, and let the third party to help you produce it by their own facility or their equipment, and take 9 months. So totally you take 1 year if we go smoothly. And after that, you already have the production data, and you already verify validation is okay. You submit a document for the commercialize, or I mean, actually, if you think about production, it means you already identify you want to make it commercialize. It means, okay, I want to provide this one become the product to the end user. So normally, if we think about everything goes smoothly, need to take around 1 month, 1 year at least. And also formulation. Formulation, they will check the biohazard. For example, if you modify something like a CGM on the biosensor, you want to have the in vivo diagnostic. It means under skin. So the protein or even some kind of allergy will be happens or not, we never know. So it will take, if you think about in vitro diagnostic, is okay. But if you think about in vivo diagnostic, it means that the biosensor is in your body. So it will take around maybe another half year for the user safety. So it means that if we go smoothly, but normally for example, like a lithium. Lithium is a very common polymer, but if the usage is too high, it becomes a toxic. So that's why you need to, normally, I already optimized this formulation should be used maybe 3%, whatever, for example. But actually, if the somebody talk to you, 3% is toxic in the body. So you need to reduce the percentage you want to use, and finally you need to reoptimize the formulation from your lab again. So that's why at the beginning, if we want to go into the production, we have a questionnaire to ask you, okay, do you do any kind of testing at the beginning or before you want to submit something? Otherwise, you need to take a 1 round or 2 rounds. It means that every round you need to take a 1/2 year. And nobody can insist going on. So that is the problem we want to reveal to everyone. And okay, for material, right? I saw the question about the material. Most of the time, if we buy the material or chemical from the supplier, actually like a sigma Aldrich. But for example, like an enzyme, sigma Aldrich is like an agent. They don't produce the enzyme. So that's why we will use original one. For example, we use the enzyme come from Japan, the biggest or the top 3 manufacturer in Japan, like Toyobo, Amano, Sekusui. Yeah, the enzyme. So that's why the enzyme is very easier to know. And actually the label scale and the industry scale, for example, industry scale or industry level were more serious, or more the criteria more tighter, and they can ensure your performance can become better. Most importantly, the price will be lower. But you need to buy a lot, because normally maybe we just buy a kilo unit, but they will ask, you need to buy 10 mega unit. Yeah. Yeah, so it's different scale, but they can ensure the quality will be very good. Actually, we are official distributor in Taiwan, one of the main enzyme supplier, because we start from the diabetes glucose strip. So we use a GOD and GDH so much. So we know who is the good and who is the best one in the world. And actually, my first career, we beat the Roche Diagnostic in Europe. The performance is the same and equal by third-party check. So the second question is,
what tips of key consideration to make like transition from large scale?
Yeah, actually, for this, normally when during the present, I would like to say, okay, we try to use out any kind of our resource, whatever which kind of spending higher or biggest the spending to solve this problem. Yes, it's correct. But finally, it's very hard to get this chemical back to our pocket, because the fuel and the expensive. So normally when we provide a questionnaire to the customer or team, we will ask, can you have the second resource? For example, if you want to use a glucose upstairs, okay, I use a Toyobo. Can you use the Amano or the another supplier? And what's the difference? Can you adjust the formulation by your own self? That is the first priority. So it's more easier to let you jump this gap. Otherwise, you will have no backup. You just be squeezed by the supplier. No choice. Yeah. So the key consideration is can we have the backup from the material, from the substrate, from any kind of the service. So for example, let's explain why our business model means we did not only sell you the product, we teach you how to make it by yourself. Finally, you don't need to depend on our service. You don't need to rely on us. So finally, you can become a friend. So we have so many alliance member overall. So that's why the backup, the second resource, is very important consideration. But normally, for example, like in academy or in university, most of suppliers they just only single line. I use this brand and no the another, yeah. But you need to provide the another choice if they want to commercialize. For example, like a regulatory, according to the quality control, the auditor, when they come, they will ask one question. Okay, you do it very well. The performance is good. So when the supplier interrupt, cannot deliver your product anymore, or even they want to close this product line and they want to have a new generation, what will you do? And during that moment, if your answer means we can wait and see, nobody can agree your submission can extend your license. So that's why that is the key consideration, the backup, everything to make some shortcut pass through prototype to large scale production easier. Yes.
Okay. Thank you very much, Dr. Warrick. And then the next question is for Mr. Jeb. So the question is, which instrument can be used as a starting point for research-based development, like to small prototype or production for the biosensor?
Okay, that's an easy question. Okay, as I just mentioned in my presentation, AD1520 is one channel. That's the most easy way to start up and most cost efficient. But let's say, as I say, of course, like a university. So for example, in university, there's a call center and then they would like to set up like some call system, then Omnia. Omnia, you can have like a nanoliter and picoliter sensor dispenser at once, right? So there will be Omnia if it's owned by university for like co-facility system. But for R&D, let's say in a single lab, AD1520 could be the best. Okay, and starting from one channel.
Okay, and then the second question is, what is the critical bottleneck in the biosensor production?
And bottleneck is a little bit difficult to answer, but it really depending on what you are doing, okay? So biosensor, I'm sure Dr. Yeni and Dr. Roy already described a lot, right? So I think for dispensing is the viscosity of the reagent. I think this is the most critical part. Let's say if you have like higher viscosity of reagent, meaning like a concentration very high, most of the time your CV will be very high. So for example, like right now, most of the reagent design or the formulation is less viscosity, below 10. That could be the best. Then for production, I think that is the key. How to keep you like a reagent deposition as accurate as possible. I think that's the key. Then by our systems is you can increase the capacity very easily if your dispensing is very stable.
Okay. Okay, Jeff. Thank you very much. So I think it's very interesting discussion and webinar about the biosensor. Very interesting, from like basic technology of biosensor and how to produce the biosensor from research to development. And also Jeff already share about instrumentation for biosensor development. So I think this is end of our webinar session. We hope this webinar can give all the participants very insightful information. And if there is still any question, you can type on the registration or email us. Then we will share the question to all the speaker.